But thanks everybody for coming to my talk today, both in person and virtually. The title of my thesis is Fine Scale Movement and Space Use of Beavers in Southern Illinois. There we go. To start out, I just wanted to give you guys a really quick outline of what I'm going to talk about today. I'm going to give you a little bit of context in regards to spatial ecology and movement, and then we'll dive into a little bit more specifically about the North American beaver. We'll move on to chapter 1, where I'm going to discuss retention time and fixed acquisition rate of glued-on GPS transmitters. And then on to chapter 2, where we're going to talk about a mechanistic characterization of space use for these animals. And then finally, I'll wrap everything up by just giving some quick conclusions. So to start off, just super broadly, my research is based in spatial ecology. And really, this is centered around the spatial arrangement and distribution of organisms, populations, and landscapes, and is specifically looking how that spatial arrangement is influencing some of the ecological processes that we're seeing. And specifically for my research, I'm going to focus on animal movement ecology and landscape ecology. And more specifically, we're going to look at where, when, why animals are moving to certain locations, and also try and figure out what type of landscape characteristics are influencing that movement and space use. And so getting in more specifically to movement, this is really the fundamental behavior that organisms have to interact with their environment around them. And this might be how an individual is accessing resources like forage or shelter in this case. This is also how they'll engage in risk or predator avoidance by just moving away from that risk. And this is also how animals will engage in any type of social interaction, whether that's positive or negative interactions. And so when I'm talking about fine scale movement, I wanted to explain exactly what that means. If you imagine that we're getting one location on an animal per day, this lets us know an area that they're using, and we can make certain inferences about why they're selecting that area versus what's available by looking at some of the associated habitat characteristics and landscape characteristics with that. But this is just one location per day. If you imagine that we take 6 locations instead, you start to get a much better idea of what's happening on the landscape. You might have an area of forage that's being used multiple times, or you might see that an individual is specifically avoiding areas that are related to risk. And then you also get a much better idea of a broader sense of what's being selected for in the landscape. So when you make inferences about what they're selecting or avoiding for, you can do that with greater accuracy. And so really, this just provides a finer resolution of the behaviors that are occurring on the landscape. And so to collect this fine scale movement data, we use GPS telemetry and transmitters. And the really classic attachment style for this are these collar transmitters. And these, you can see examples here for elephant, elk, white-tailed deer, bobcat, and coyote. But we also have these backpack style transmitters, commonly used with birds. And then we also have glue-on transmitters used with tortoise or with this really cute fair seal, ear tags for bison. And then there's many species that get specialized modified attachments like with this giraffe so that we are able to track their movements. But really, GPS telemetry has come a super long way in the last 30 years. We've increased the battery life of these units. We've decreased the size, and we've made them much sturdier so we can track a wider array of species and in a variety of different habitats. Now, that being said, there are still some individuals that are difficult for us to track. And semi-aquatic species really provide some unique challenges with using GPS transmitters. And so really what causes a lot of the issue is they have this fusiform shape and tapered neck, which really is just unsuitable for collar style attachments. It's just going to fall off more than likely. And furthermore, anything with a strap like a backpack or a collar style attachment could just be chewed off. So you're limiting the time that that animal can be tracked. And finally, it also presents a little bit of risk because if you have straps on an animal that's submerging in the water, they can potentially get hung up on debris and drown, which we 100% would like to avoid. But also you have problems with GPS fixed success rate. And this is related to the habitat characteristics associated with these species. Oftentimes they'll have a den or a lodge or a burrow, something of that nature they're staying in during the day. And GPS signals are not able to propagate through that substrate to get an actual location. You run into the same issue with all the dense vegetation associated with these areas, blocking satellite signals. as well as if the transmitter is submerged, it takes just about an inch of water for a GPS transmitter to no longer be able to communicate with satellites. So based on all these different challenges that are associated, it makes these animals just really difficult to apply GPS technology to. And so the theme of the talk today, the North American beaver, is one such species that provides some really unique challenges. And so to give everybody a little bit of history about beaver, historically they inhabited pretty much the entire lower 48. They were absent in Florida because of the predator density with alligators there, and they were absent from some of the more harsher desert environments in the West. But otherwise, ubiquitous across the lower 48, they encompassed much of Canada and all the way up into Alaska. Unfortunately, around the 1800s, some really heavy trapping and harvesting led to the extirpation of this animal from much of its eastern range and other portions of the range. But fortunately, in about the 1900s, wildlife managers started to implement some harvest and trapping restrictions to give these populations just a little bit of a breather, let them recover. And they also started translocating beavers back into areas of their range where they had been removed. So they've actually recolonized pretty much most of what they used to inhabit, and they've proven highly adaptable despite all the anthropogenic changes to the landscape. And beavers play a really huge ecological role. They're considered both keystone and keystone species and ecosystem engineers. And this is primarily due to the damming behavior that they're very well known for, which creates these pond and wetland complexes, as well as their selective foraging really has a huge influence on the vegetative structure. And this really benefits a number of different flora and fauna. And there's also been some recent research showing that beaver presence in an area actually helps increase climate resiliency at the landscape scale. And this is because in areas with like drought and wildfire, beavers, when they're present and building these dams to create these ponds, wetland systems, they're retaining more of that water late into the dry season so that water is still available for the plants and wildlife there. And then in addition to this, maintaining some water in both the soil and the vegetation makes these pockets a little bit more resistant to burns. So a number of ecological benefits for having these species around. And real quick, there are a couple of specific fundamental behaviors associated with beavers I wanted to point out. They're what's considered a central place forager. And this is just a really basic concept that means they have the central place that they start out from. They'll leave from there to go on a foraging trip and then return to that central place. And the really classic model for this is a bird, leaving their nest to forage and coming back to that nest. And in addition to this, beavers are highly territorial. They invest so much time into constructing these dams and these lodges that they want to protect those areas as well as the resources that are in close proximity. So they will go kind of to the periphery of their range and deploy these scent marked mounds, which are just castor mounds, and they're just scraping up mud and sticks from the bottom of the water body they're in, piling them there on the edge of it, and then scent marking that. And that's just to communicate to other individuals like, hey, this is my area, stay out, don't mess with me. But both of these really result in some highly structured movement that would be really interesting to look at in more detail if we were able to collect fine scale movement data for these species. So traditionally, the way these individuals were tracked is with what's called a VHF transmitter. And this just emits a signal that can be tracked using an antenna. And this does work to get locations, but some of the issues with this method is it's very logistically demanding to get multiple locations on an animal, especially, just to get one a day, let alone multiple locations per day. And the other problem with this is if you're constantly present in the environment that the animal is, you're running the risk of potentially altering their natural behavior, which defeats a bit the purpose that we have of trying to monitor what they're doing in their environment. And so there have been some attempts to track beavers with GPS transmitters, but again, all the challenges I highlighted for semi-aquatic species obviously apply here. And so many of those attempts have been abandoned due to poor performance. Alternatively, they've just tracked them for a really short period of time to try and avoid some of this. But ultimately, if you do that, you're missing out on some of the long-term behavior that's occurring in the landscape. And so the motivations for my research were really pretty simple. There's just not been several attempts to actually study these species with GPS technology. And given all their ecological impacts, it would be important to understand their fine scale movement behavior. And they also have economic implications. Industrious as they are, this can result sometimes in some human-wildlife conflict situations. They might dam a culvert, causing some road damage, potentially flood agricultural crops. So we want to be able to mitigate that, but this also, having a better understanding of how they're using their space also presents the opportunity for us to inform some of those translocation and reintroduction efforts that are going on in areas of the West that would benefit from beavers being present. So the main goal of my research is we wanted to just investigate the fine scale movement and space use of beavers and try and identify the mechanisms that were driving home range structuring for these animals. And then in order to do that, obviously, we had to have a secondary goal where we developed and tested these methods of GPS transmitter attachment. And we wanted to identify which was going to give us the greatest retention time. So really just how many days did the transmitter stay attached, as well as which had the best GPS fix success rate. And so that's just what is the percentage of actual fixes that we got versus what were attempted. And so the study area where I conducted my research was located in southern Illinois. It's called the Union County Conservation Area, and it's about 25 square kilometers, owned and managed by IDNR. And I think they acquired it around the 1940s, primarily as an overwintering site for waterfowl. But obviously there's a ton of other species down there, and there's certainly an abundance of beavers. So a great place for me to conduct my research. And so moving on to fixed acquisition rate and retention time of glued-on transmitters for these animals. Some of the questions and objectives associated with this chapter were, we wanted to see which GPS transmitter attachment method provides the greatest retention time and the best fixed success rate. I also wanted to see how that retention time and the fixed success rate were affected by things such as the age, sex, or the season. And then we also wanted to see if we were collecting a sufficient amount of data with these methods to actually get at biologically meaningful behaviors. And so in this case, we really wanted to see, are we capturing home ranging behavior with the amount of data that we're collecting? So moving on, I trapped these animals using cable restraints. And you can see that these were designed really to target specifically beavers. We didn't have a single instance of bycatch. We had a ton of things like otters and other little critters cruising through these all the time, and never had any issues. But I want to stress that this was a safe way to capture these animals. Down in the bottom left, you can see a really good example of a beaver being caught by a snare. They're going to get their front arms through, and this is basically just going to cinch down around their waist like a belt. So really great way to capture them, pretty safe method, and we had a lot of success with this during the course of the trapping seasons. And so the three different attachment methods that we were testing, we started out, we wanted to kind of follow suit with that traditional VHF attachment method since the retention time was so high. So we tried putting some GPS transmitters on the tail, and you'll notice that I also have a transmitter glued to the back. This is not a GPS, this was just a basic VHF transmitter. And we wanted to glue one of these on just so we could get a one-to-one comparison of the retention time between the two methods. And then we also had the upper back and lower back GPS mounts. So moving on to how many I actually deployed. In the spring, I had 3 tail mounts that we deployed, and then 16 lower back mounts. And then in the fall, I deployed 7 upper back mounts and 6 lower back mounts. And I get that's a pretty wonky distribution, and so I'll explain really quickly. We initially planned the study, and we were going to try and deploy 8 of the tail-mounted transmitters along with 8 of the lower back transmitters in the spring. But unfortunately, after 3 deployments of the tail method, we found it was not working. We had a fixed success rate of less than 1%. So essentially, you're not even getting any data on the animal. That's worthless. And we wanted to abandon that. We didn't feel there was any need to prove the point further that that didn't work. So for the rest of the spring, we shifted everything to the lower back mounts. Of the 3 transmitters I did deploy on the tail, we had 1 unknown mortality. 1 transmitter malfunctioned, and I was unable to recover it. And then we had 1 individual we were able to recapture and remove the transmitter. So for the spring, again, we had these 16 lower back mounts. I did have 1 transmitter malfunction in this group, And then we also had 3 mortalities. This included predation by bobcat, 1 unknown predation, which honestly almost could have also been a negative interaction with another beaver. There just wasn't a lot left by the time I got there. And then we had 1 unknown mortality. So given we have these malfunctions and mortalities, we wanted to remove these from any further analyses since that didn't give us a true representation of retention time. So I ended up just keeping 12 for the actual analyses. And then in the fall, we put out the 7 upper back mounts and 6 lower back mounts. Fortunately, no mortalities this time around, but we did have 1 more transmitter malfunction that was a lower back attachment. So again, we saved 12 for analysis. So moving on to some of my results, the only thing that did not affect retention time was the age of these animals. And so attachment, season, and sex all affected the actual retention time. And so if you look at figure A, on the x-axis there, I have LF, LS, and UF. And this is just the different attachment method groups we had. It includes lower back fall, lower back spring, and upper back fall. On the y-axis, we have the retention time with respect to days. And we found that the lower back fall method provided the greatest retention time, followed by upper back fall, and then lower back spring being the least efficient method. During for season, we found that fall had the higher retention time as opposed to spring. And for sex, we found that males had a higher retention time when compared to females. And moving on to GPS fixed success rate, the sex and age had no impact on these results, but attachment and season did. The lower back fall, the x-axis again is the attachment methods, lower back fall, lower back spring, and upper back fall. And then the y-axis is indicating the fixed success rate in terms of percentage. Both the lower back fall and upper back fall were the top performing methods. These weren't statistically significantly different, so both did pretty well. Lower back spring, again, was bottom of the pack in terms of performance. And then for season, it follows that fall had a higher fixed success rate when compared to spring. So before moving on and talking a little bit about were we able to identify home ranging behavior, I wanted to talk about a bit what that is exactly and how we actually go about identifying it. And so one way to think about home ranging behavior is an animal exhibiting restricted space use and range residency. And so if you imagine you put a transmitter on an animal, you might only be capturing a certain limited amount of space for the first day. But over time, the amount of space that you're capturing is going to increase. And you'll continue to see this happen until you eventually reach a point where it's going to start leveling off. And once the variation in space use levels off at this point, and this individual is displaying restricted space use, and this is what we would consider home ranging behavior. And so one of the ways we can actually identify that is with what's called a variogram. And there's a couple things I'd like to point out. On the x-axis there, it's time lag in days. On the y-axis, this is just the variation in space use. The black line itself is indicating your empirical movement data, so the actual locations you collected in the field. And then this red line is a model that's fitted to the data. And so there's one point that I would really like to highlight for these, and that's where this begins to flatten off or where it reaches its asymptote. This is basically the point at which the individual is starting to exhibit that restricted space use and home ranging behavior. And you'll note that as this continues, it stays flat throughout time. So over those extra time lags, it's using the same amount of space, and this individual is basically showing home ranging behavior throughout that whole time. If that graph just continued to increase and didn't actually flatten out, then you don't have enough data collected to get home ranging behavior, or you might just be capturing dispersal or migration of that species. And so ultimately, we use these variograms to visualize the autocorrelation in our movement data. And more specifically, this model, the red line that's fitted to the actual data, we can derive the estimate from that asymptote, extract that value to basically say, hey, this is the amount of time it took for this individual to display home ranging behavior. And then based on some of this, we were also able to calculate, make some really accurate home range estimates using autocorrelated kernel density estimation. And so a couple of examples for this output. On the right, you can see some home range estimates for Scar and Pumbaa. But more importantly, I'd like to point out the variograms to the left. One thing that you'll note is the model that's fit to the data there reaches its asymptote really, really quickly for these animals. And then also you'll notice something unique is these spikes or oscillations in the data. And this is really just showing you the difference in the daily activity periods for a beaver, really active at night, using a little bit more space, and then during the day, remaining in their lodges, and so obviously less use of space. Moving on to some of my results for this. In figure A, you can see on the x-axis there, we just have all individuals. On the y-axis is the asymptote, time it took for an individual to reach that point in minutes. And our results ranged from about 20 minutes up to about 540 minutes and averaged around 100 for these individuals. And then in figure B, on the x-axis, we just have the seasons, fall and spring. And the y-axis is the AKDE home range estimates in terms of square kilometers. And we found that spring had higher or larger home range estimates than in fall. But I'll note it's not much. I mean, these are mostly all under about half a square kilometer. So these are some really tiny home ranges that they're using. And so some takeaways from chapter 1. Again, the lower back attachments in fall were by far the most effective method. And I think there's a couple of different things that are actually going on here that explain that related to the biology and the behavior of this animal. So during the spring, they are going to have kids present, and they're going to have to allocate a lot more time to parental care, which means more time spent in the lodge, as opposed to in the fall when they're really no longer required to provide that parental care as much. And they're going to increase their foraging to collect food and construction materials for the winter. And so just by virtue of being out of the lodge that much more in the fall versus the spring, you're going to get a better fixed success rate. And then in terms of retention time, it kind of goes hand in hand. The thing we noticed on all these transmitters that we recovered is they were all, heavily scarred up from what appeared to be a lot of chewing. And furthermore, most of the times where they were located after being dropped was actually inside the lodge or den or within about 10 meters. And so I think the thing that's actually driving retention time most is the social grooming these animals will engage in. It seems like they were just chewing on the attachments and one beaver did not want it on another one. So they were pretty adamant about getting it off there. So a little bit more time spent in the lodge in the spring would probably provide a little bit more social grooming activity. So that could contribute to the lower retention times we saw. But this also overlapped with the time period of the year that beavers are engaging in their annual molt and shedding some of their winter coat. And so the combination of the social grooming and then shedding some of the fur that the transmitter was actually attached to probably resulted in that lower retention time that we got during the spring. Now, I also want to highlight too that males had a greater retention time than females. Males do have a little bit higher propensity to engage in some territorial behavior. They might have a little bit longer activity periods and things of that nature, whereas females will engage in a little bit more parental care. So it could just be a factor again of females might be spending a little bit more time in the lodge versus males and subject to a little bit more of that social grooming that's going on. And we were also able to capture this home ranging behavior for 21 of our 24 individuals, which was really good. And I just want to note again too, we had that really, really short time compared to the example variogram that I had up. And there's a couple reasons for this. Another characteristic of restricted space use is an individual's ability to cross their home range several times. So because those home ranges were so small and often inundated with water, beavers were able to traverse these areas very, very quickly, resulting in that quick home ranging time that we found. And finally, I just want to point out again, the tail-mounted transmitters did not work. I would avoid this at all costs. And the reason the fixed success rate was so bad is because these likely stayed submerged the whole time they were attached to the beaver. And again, like I said, in the context, it only takes about an inch of water to prevent that GPS satellite from making a connection there. So again, that's probably why it didn't work very well, but would avoid that in the future. So moving on to my second chapter, mechanistic characterization of space use. So the questions and objectives we had here is we wanted to see what behavioral mechanisms are driving patterns in space use for these animals. And how is that influencing home range structure? We also wanted to see what factors related maybe to like environmental characteristics or individual level characteristics like sex and age were influencing the space use and home range structuring. And so traditional approaches to evaluate movement data are often only focused on the time spent in a location. And one of the common methods used to look at this is called a utilization distribution. And this is just a really simple method where it's just looking at how intensely certain parts of the landscape are used as opposed to others. So areas in the red indicate high intensity of use, and areas that are characterized by this blue color are just low intensity of use. We also, a common tool for wildlife biologists are these resource selection functions. And so basically, we're looking at the time spent in specific areas, and then the habitat or resources that are associated with those to see what an animal might be selecting for versus what's available on the landscape. The issue with these approaches is you're just looking at the intensity of use for a location, and you might be missing the functional role that location is playing for an animal. And so, for example, if you think about an animal at a rest site, such as in this example, they may only visit it a few times, but each time they visit it, they're going to spend a lot of time there. And ultimately, the same intensity of use could result from a fundamentally different behavior. And in this example, I have an individual that's visiting a profitable patch of forage several times, but for shorter durations, but again, ultimately resulting in the same intensity of use. So without looking past just the time spent in an area, we might muddle a little bit our inferences on the value of these locations for individuals. So how are we actually going to uncover these mechanisms that are driving space use? Where these traditional approaches are looking just at the time spent in the area or a total duration, they're really missing that this is a function of several other movement properties. And this includes the average duration of those visits, the total number of visits, and the interval between visits. And what would be nice is if we could take these 4 movement properties and combine them into a single representation of space use for an animal so that we get a better idea of how they're using the landscape. So I developed this approach called UseScape, and I'll walk you guys through it really quickly. Basically, beginning with A there, you're going to take your raw movement data and convert that into a trajectory so that we know how these locations occurred in time. And then we want to overlay that trajectory to a grid. In order to determine the pixel size for this grid, we just use GPS error of our units. And then once we get the trajectory on the grid, we can look at each pixel and in terms of these little blocks of space they're using, and we know when an individual has entered it, how long they spent there, when they left, and when they returned. And then based off of this, we can extract all these different values that we wanted to look at. So again, we have frequency or total number of visits, total duration, average duration, and average interval. And once we get all these values for these different pixels, we can use machine learning to cluster similar homogeneous types of space use and basically get an output similar to what's here, where you get maybe 5 different types of space use for these individuals and where they're located on the landscape. And I'd like to mention too really quick that we did this in a 2-step process. So we did this for each individual, and then we took the average across all individuals to get a general population level space use. And then once we had that general population level space use, we then reapplied that to the individual level. And so we used 15 individuals for this analysis. Basically, the requirement was we needed at least 20 highlighted pixels for them to be included. And this is just because anything less than that, they were a bit data poor and they weren't adding much to what we were trying to figure out here. And so this is an example of a conversion of all the raw data with the associated home range boundary into an actual UseScape figure. And so, like I mentioned, we did that population level clustering. And so we get a set of values for this, which is indicated in this table here. And this can be a little bit much to look at first, so I want to walk everybody through it. Along the very top row, each column represents a different intensity of use type that we've identified. And then along the left side there, we have the metrics that we were looking at. So total duration, number of visits, mean duration, and mean interval. And more specifically, just to make sure we're clear in reading this, if you look at this top row, you can see that total duration is indicated for each of these different intensity of use types. And each of the values that I have listed here are relative to one another. So the two pluses that are in lodge use indicate a really, really high duration, whereas the minus in the low use indicates low duration, and zeros are more of an average. And so moving into each intensity of use type, again, we were able to highlight these lodge and den use areas, which is a really key area for these animals. And you'll notice that they spent a lot of time here, visited it often, and if they did leave, the interval to return was pretty low. In addition to this, we identified these two medium use clusters, medium use short duration and medium use long duration. And something I'd like to note is you'll notice that total duration for these was very similar, but this arose from a combination of different movement properties. In the case of medium use short duration, we had a high number of visits and an average mean duration. Whereas in the medium use long duration, it was just an average number of visits with a higher average duration of those visits. And then moving on, I had two low use clusters. They do differ a little bit in total duration, but I'll admit when I was converting the values over to this, the value for low use irregular just barely made the cut to be a 0. So that's a very low average. But the big thing I would like to highlight here is both of these are low use, but they have clear differences in the interval that's associated with them. One is regularly used, whereas the other one is just very rarely used during the course of our tracking period. And so then we wanted to see what was influencing the occurrence of these different types of use on the landscape. And so we wanted to evaluate this with respect to distance to the home range edge, whether or not a pixel was wetland or not. And then we also wanted to know the distance to the nearest lodge or den. And we got distance to lodge and den based off of the clusters we were able to highlight. We did confirm that these were lodges and dens during field work through ground truthing efforts. But then we wanted to see how the response to these variables may be varied with respect to age and or sex. And then we compared all of these used areas and those associated characteristics with what was available on the landscape. And so in other words, all this gray that's around the used areas. So moving on to some of my results from that, we used a mixed effects logistic regression and found that the top model for lodge use was a combination of sex and age and the environmental covariates we included. And you'll notice on the table, anything that has like multiple pluses versus 1 is just indicating the intensity with which they responded to this variable. And so for both or all adult male, adult female, and non-adults, these clusters were more likely to occur away from home range edges, but that was a more intense response for adult males as opposed to, say, non-adults. The top model for our medium use short duration cluster was an interaction between sex and our environmental covariates. And you can see that males, this was more likely to occur for males away from home range edges, close to the lodge, and it was positively associated with wetland. And it was a similar response for females, though less intense. And then the top model for the medium use long duration cluster included an interaction between sex and the environmental covariates. And for this one, it was much more likely to occur for males away from home range edges, as well as in close proximity to the lodge or den. And for females, it was just more likely to be found in close proximity to the lodge. For the low use regular cluster, top model was an interaction between age and the environmental covariates we were using. So adults, this was more likely to occur away from home range edge, and it was positively associated with wetland. And for non-adults, again, more likely to be found away from home range edges, though more intense response, and likely to be found in close proximity to the lodge. And then finally, the top model for the low use irregular cluster included an interaction between sex and the environmental covariates we included. And this one, for males, it was more likely to be found away from home range edges, and for females in close proximity to the lodge. And so moving on, we have these 5 different types of use that we highlighted. And so again, one of the goals is we wanted to see how this was influencing home range structuring. And so we found that for most beavers, on average, they would have about 2 to 5 lodge or den sites within their home range. And these were generally associated with the core of the home range located away from home range edge. In addition to this, we were able to highlight these two medium use clusters, which were generally located near the lodges and away from home range edges. And because of this juxtaposition to the lodges and their relative high use and low interval, I think that these are actually resource patches that these animals are taking advantage of on the landscape. But there's a very key difference between the two of them. The medium use short duration cluster, if you'll remember, was positively associated with wetland, whereas the medium use long duration was not. And so likely what the difference is here is that medium use short duration is representing areas of forage located in water as opposed to areas of forage on land for the long duration. And this would explain the difference in how those were used. Areas in water are just going to be more easily accessed for beavers, whereas areas on land are going to take a little bit more time and energy for them to actually profitably forage on. And so we also had this low use regular cluster. And even though this was low use, it seemed pretty important on the landscape. It was the most prevalent cluster across all individuals. And it really seemed to indicate their preferred movement paths to access shelter and resources. And really kind of signaled some very directed, repetitive movement between these areas. And then finally, we had this low use irregular cluster. And this just seemed to represent maybe some excursions from the daily routine. They were so rarely used that didn't seem like, it was not a regular occurrence. And it could have just been they picked up on an unfamiliar scent they wanted to check out, possibly investigate a new profitable patch of forage, or things of that nature. But something I would really like to highlight here is pretty much everything was generally associated in the core of the range and away from home range edges. And it seemed like they like to leave a bit of a buffer with the space they were using. So my idea is that this is probably just a way to avoid any type of negative territorial interaction with a neighboring colony of beavers. And so again, chapter 2 takeaways, we were able to highlight these 5 different types of use for this population. And again, something I want to highlight is we were able to delineate between these two similarly used areas. And if we hadn't looked at it in this way, these probably would have been lumped together since they had similar total durations. But we can see that specifically between the medium use areas, you have a fundamentally different thing going on there where there's foraging occurring on land as opposed to in water. And because the medium use short durations are getting higher number of visits, the resources there are probably being depleted quicker. And so this has implications for when a beaver might deplete those resources completely and might need to shift their home range. And then we also had these low use clusters, which otherwise would have been grouped together. But one is playing a very critical role on how those animals are actually accessing resources on their landscape, whereas another one is just representing these kind of random exploratory behaviors they may engage in within their home range. And then finally, again, we were able to make inferences regarding home range structuring for these animals. And again, it's basically a lot of highly structured movement for the 4 main groups there, with the weird excursions that happen occasionally, and maintaining that buffer away from home range edges. And so to wrap everything really quickly with my conclusions, some conservation and management implications. We were able to identify a GPS transmitter attachment method that was capable of collecting 1 to 2 months of data. And this was sufficient for capturing biologically meaningful behaviors. Here we looked at home ranging behavior and home range structuring. But because there's so little research that's been conducted on these individuals with fine scale movement data, this will open the door for other researchers to try and dive in and maybe ask some other questions now that they have the methods. And then we were also able to get this deeper understanding of home range structuring and space use.
Because they're on Netflix.
And what would be really nice is I think this could be applied to some of the translocation and reintroduction efforts that are going on. If we have a better idea of how they're using space, we can probably identify locations for translocation with greater accuracy. And then we can also understand very specifically how they're going to impact that environment once they're put there. So again, understanding the rate at resource depletion for certain patches that they might access, and then how core home ranges may shift from there. And the really big takeaways from this research is I want to drive home the point that it's important to understand patterns in space use, but we really want to understand the behaviors that are driving those patterns as well, because that can tell us a lot more about the functional role a location is playing for an animal, and that would have, certainly have implications for conservation and management actions that we might take. And again, delineating between similar types of use is just not possible if you're just looking at the intensity of use in area. We really want to look at all those movement properties at once to get as good of a picture of what's going on in the landscape as possible. And then I do also want to stress that this is not just a method for beavers. This has implications for all wildlife. And in fact, as a lab, we're working on applying this method to elk, Galapagos tortoise, coyotes, and white-tailed deer. So, as the world, constantly goes through rapid changes, climate change, landscape change, anthropogenic development, it's going to be crucial for us to know how animals are responding to their environment around them. And this will give us, again, a much better idea of how to inform these conservation and management actions that we might take. Hopefully this will be in review while I'm in a race to beat them to get my second chapter published. But yeah, with that, I'd just like to say thanks to Dr. Guillaume Basteau-Rousseau for securing funding through SIU for my research. I'd like to thank the staffs at IDNR, Forest Service, Kenneth Delahunt, Drew Fehrenbacher, and Craig Bloomquist were all super helpful in this process. I'd like to thank Telemetry Solutions and Quentin Kermine for being daring enough to at least build a transmitter for us to try this. I'd like to give a huge thanks to my advisor, Guillaume, as well as my committee members, Charlotte Narr, Clay Nielsen, and Marge Brooks for all their help along the way. I'd like to thank my technicians, Kristen Botzett and Blake Apt, the awesome people in the Spatial Wildlife Ecology Lab and Cooperative Wildlife Research Lab, and then also my friends and family, especially parents JB and Becky, siblings Natalie, Daniel, and Kaylee, and grandparents Ada and Brooks. And then I would like to give a special shout out to all the beavers who willfully participated in this study. And with that, I'll open up for questions.